Hello, everyone. Welcome to session 2 of this year's BGA. I'm Damon. I'm the organizer for this year, and with me today are Will and Yumi, who are all three of us are the developers of the TreeVal pipeline. In the chat, there is there's a form that I'd ask everyone to fill out, just so that I can get a good handle on the people that are joining these sessions. And the code in this form is simply just TreeVal. Feel free to do that now or wait till the end of the session. It's up to you. But yeah, that's it from me. If you two want to take it away now.
Yeah. Can I have the share?
Of course you can. Yes. Stop share.
Okay. Hello everyone, welcome to the BGA24 presentation on our Nextflow pipeline project, TreeVal. This project was developed by myself, Yumi, who will also be presenting today, and of course colleague Damon, who is facilitating. This slide deck was actually originally put together by Damon. Everything shown here today will be available on the TreeVal repository and the wiki if there's more you want to know, so head over there. Here is a rough itinerary of the day. First we'll walk through TreeVal, then demonstrate it, followed by a short break. We will also have a look at uploading our outputs to JBrowse and HighGlass so you can see the final product. We'll also finish off with a quick run through of the related curation pretext and Zippy pretext tools. If you have any questions, please put them in the Discord channel. We have a 2-hour slot, so we should have absolutely plenty of time to try stuff out for yourselves. So first, I should try and explain what TreeVal is. The goal of the Tree of Life Evaluation TreeVal project is to generate the data required for the curation of high-quality genomes. Curation in this context being the manual rearrangement of the genome assembly to improve its contiguity. The TreeVal itself replaces a number of previous tools, including a SnakeMake auxiliary pipeline and GVal, which served many of the previous projects run by the Tree of Life. If you're more interested in the curation side, by the way, you should sign up to attend or at least watch the recording of the GRITS talk, for which Damon is, what's it called? The manual curation?
Manual curation talk, yes. I will find the date and time for that and put it in the chat.
Thank you. Yes, that should all have more information on the manual curation process that TreeVal is designed to support. TreeVal itself is being used within the Tree of Life project for the last year and a half, and over 1,000 genomes have already gone through this process. Here's an example of visualization of the data outputted by our pipeline. This presented in JBrowse, which is a web browser-based JavaScript-based genome browser, which we'll go into more detail on later on. JBrowse is more powerful than we're currently presenting here and able to support lots of data types. So, the TreeVal project. Sanger, especially the Tree of Life program, is currently making a push into standardizing the larger pipelines and converting them into a singular language, Nextflow. Nextflow DSL2 is a dynamic pipelining language built on Groovy. This comes with a variety of benefits related to training time, modularity, scalability, and being largely hardware agnostic, relying only on compute environmental configuration files to make use of HPC systems like LSF and Slurm. As part of this standardization, we'll be using NF core standards mixed with some homebrew practices that are still currently being written. We wanted a simple front-end browser that could be easily navigable. As this, we settled on JBrowse. One of the advantages, sorry, one sec, I will just check. Yeah. Just for context, the TreeVal project was designed to replace the existing GVal project, which was used by the Tree of Life up until a few years ago. So the main scope of the TreeVal project was to replicate GVal initially and then to extend for future use. One of the advantages over GVal came from moving towards flat file formats, reducing the storage requirements, and then also making it easier to share and output, making it easier to share the outputted data. As our initial goal was the replication of the GVal pipeline, we initially stuck to close to GVal as possible, only diverting as and when we came across an issue that could only be fixed with an alternative. One example of this was moving from Minimap2 to Miniprop for the peptide alignments and using our own version of the self-comp algorithm. These workflows are designed to be extendable within and share within the community. So if anyone has any ideas for how to improve on existing sub-workflows, please familiarize yourself with them and look into creating an issue on our GitHub page. Okay, up to running TreeVal. This is the sample YAML, which contains everything needed to run TreeVal. As you can see, so yes, not every single one of these properties is currently used, but they are useful for tracking information about that particular assembly being analyzed. Though, and there's also some scope for future use as well. Oh yes, just for context as well, the gene sets that we use have to fit these particular particular file structure, which we won't go into today, but is shown an example of which is shown on the right-hand side. I'll just also quickly mention that we have a tool for validating the input YAML. It's called FASMAN. It has a number of TreeVal related functions, but the one that you'll be interested in is the validation of the YAML, which we have an opportunity to look at later. Okay, so now for a quick overview of the TreeVal pipeline. Here is a simplified diagram showing the inputs, the general workflows, and the output files, and where the output files are visualized. There are two basic entry points we use. We run either in rapid mode or in full mode. Rapid mode is the ones highlighted in purple here. The vast majority of our assemblies run through TreeVal are run in rapid mode, though but full mode is used for some more complex genomes, for example, birds, or can also be done on request by people in the manual curation team. You can see that TreeVal is quite large at this point and produces a variety of file formats. Everything can be viewed in JBrowse aside from the high glass specific mcool and the pretext file pretext viewer. But quite interestingly, JBrowse has actually got quite a good documentation now. So if there is any additional file types you'd like to display in JBrowse, it is possible to develop plugins to do so of your own accord. These sub-workflows replicate the functionality of GVal. The only additional new one from GVal is the ancestral element analysis sub-workflow, which was originally developed by Charlotte Wright from the Blackster Group and added into our pipeline. It allows for the mapping of putative ancestral chromosomal units using BUSCO genes. Currently, this is only available for Merian units for Lepidoptera, though we are looking to expand this to other clades. Simply, the details of this are found in the project. Now we're going to go into a little bit more detail about some of the individual sub-workflows, which I'm going to hand over to Yumi, if that's okay.
Yep, thank you. Could you stop sharing? I would like to share my screen. I hope you can see my slides okay. Yes, my name is Yumi. So I'm one of the developer of the TreeVal. So just thanks Will to give an overview of the entire TreeVal workflow. And I'll walk you through the main workflows including in the TreeVal. So I would like to break down into two parts. And first, we'll go over the workflow used for the rapid curation, as Will just mentioned. So there are 5 workflows actually we include for rapid curation. And then we will dive into the workflows used for full curations. As you can see, and the first important workflow is the Hi-C mapping. It is the most crucial steps in our genome correction process, as I so allow me to say that. And if you have experience already with the manual curations, maybe this concept is easier for you to understand. And for those people who haven't used a Hi-C map before, and I would like to emphasize this, the importance of the Hi-C mapping is one of the most critical steps in the genome curation, because the overall presentation of the Hi-C map gives us a direct reflection of the quality of the genome assembly. Our approach involves using Hi-C map alongside with other evidence to fine-tune the existing Hi-C map, aiming for a clean, diagonal only as much as possible. So that's the ultimate aim. Although most of the genome cannot reach to that state, but we try our best and to utilize all the evidence we have. Here's our workflow works. First, we align the Illumina Hi-C read. So as you know, Hi-C read, they're essentially parent Illumina read. And so we align the Hi-C read to the reference FASTA produced by the assembly pipeline. We strictly require the input to be indexed CRAN files. Why? So as you know, CRAN files, they are container-based. And so there are 2 main reasons. CRAN files are much more storage friendly, saving space compared to BAM and FASTQ files. And secondly, because our alignment process is handled by 2 supermodules, I will go through it in a minute, which take care of the heavy lifting. After user pick up the aligner, either Minimap or BWMM, we index the assembly and fit both of the Hi-C reads and indexing into the supermodules. So the supermodules, and yeah, now you can see the 2 boxes highlighted and in the graph. And as its name suggested, use a CRAN filter developed by James Bonfield in Sanger. And to split the CRAN files into manageable chunks, we typically process batch of a 10,000 container Hi-C read, which makes everything much more resource efficient, also allows for parallel processing and reduce the risk of the index. So that's very important. So many people try to use smaller batches of containers that go through Hi-C mapping, but end up with tons of index. So that's not very friendly for our curation work. And next, we align the batch containers and filter unwanted noise. And once that's done, we use the SAM2 fix mate to make sure the mate information for paired and read is properly set for both reads as a pair. In the final step, and we merge everything using SAM2 merge. As you can see, so SAM2 plays a major role in here, thanks to Hughley. And I'd like to just point out a little feature we have added recently, YAS. So I put down the reference at the bottom here. So YAS represents as yet another Hi-C scaffolding tool, which is developed by my colleague, Tensi. So what we use YAS for, we now use YAS to generate binary alignment files, allow us to store the current alignment results, whereas only retain the coordinate of the alignment. And why we have to save alignment of current state at the moment. And I will save it for now, and I'll explain it later. And then we generate a pretext map based on the merge BAM in both normal and high resolution for easy visualization. For users who prefer use high-C files, yes, we have this part covered too. And to prevent user from hogging too much memory, and we control the input BAM size and with a sum to subsample function. So as you can see, so samples everywhere, and keeping under only 50 gigabyte. On another branch of the workflow, and we use a sum to mark dupe and to mark up the duplicate in the merge BAM file, then generate contact pairs from the data. So these contact pairs are used to create high resolution, high class, and cool files, which allow us to visualize everything clearly in high class. So this is a high-C mapping. And as I said, if you have any question, feel free to drop your question into the Discord. And the next
one, sorry, related
okay.
to the previous sub-workflow from Sam. Why are we subsampling the BAM? If it's over 50 gigs, does it not reduce the density of the contact map? Is this to get around issues with mark dupes?
So we thought about to maintain the user high-C file in the original size, but for our production at the moment, because we produce tons of assembly metrics every day, so as you see, the user high-C taking up a lot of memory and a lot of timing as well. So for the current approach, we only allow the 50 gig. Yes, you can take it out, and you can take it out the intermediate file and to produce the high-C map with a full size of BAM if you wish. And in the future development, we still have to drop back. So we have this part implemented already. We could make it options, and with the full size of a merge BAM to produce user high-C or the subsample version. So that could be optional in the future. Is that okay? Yep. So I'd like to move on to the next important workflow, which is the read coverage. And it's one of the key metrics we use during genome curation, as it gives us a lot of valuable insight into the quality of the genome assembly. It helps curators spot important features like haplotypic typification, gene duplication, and even big structure difference like sex chromosome or big B chromosome. By looking at the read coverage, curator can just jump on the problematic area, check out well, and the assembly is holding, how's the assembly holding up, and get a better feel for the overall structure of the genomes. In the latest version of the TreeVal read coverage workflow, and we have made some pretty big improvements. One of the coolest things is that it can now handle multiple types of sequence read. Whether you are working with a PacBio COR, I believe some of you are still using that. And PacBio Hi-Fi, yeah, is more than one. Even PacBio CCS, and those being corrected quality read, and Illumina short read, and also ONT from from Oxford Nano. And these are all including in our read coverage, and the workflow can handle them all. And all you need to do is just tell what kind of read you are working with in the input YAML, and then the TreeVal take care of the rest, adjusting the alignment setting accordingly. For the read alignment, and we use Minimap again and developed by Damon-Lee, which is an important part of calculating read coverage. After aligning the reads to the reference genome, the workflow gathers all the back files that were generated, that is sorted and organized everything to make sure the right format is in the right format. Once that's done, we use a back2 genome cut to produce a sorted back file along with the chromosome size file. It gives us a detailed coverage back file that shows how well each region of the genome is covered by the read. And to make all this data easy to work with and visualize, and we convert the back file into BigWig format. And BigWig format, yeah, might be most of yours and favorites, is super handy because they let you quickly browse through the data in a popular genome browser like UCSC, HiGlass, and IGV, and JBrowse, and the genome browser I developed a long time ago, Darwin browser, which so it's a very handy kind of a data type. And a standout feature of our read coverage workflow is generate a bunch of punch list. These files are essentially report that points out interesting spots in the coverage data, like the area with a half coverage, zero coverage, and super high coverage. These are crucial for curators, as they can quickly focus on parts of the assembly that might need attention. For example, a region with no coverage could signal a gap in the assembly, while a spot with a usually high coverage might mean there's a duplication or an assembly's artifact. So punch lists make it easy for curators to call on those areas, which really speed up the curations process. And so this is the read coverage. If you have any questions, so you can ask me right now, or just leave your chat there, and I will go through after I finish my presentation. And so third workflow is repeat density, and this is the one of the most important one for rapid curation. And repeat density analyze so it's a very important method, and it's used to scan low complexity repeat region in the genomes. And this process helps curators to gain a quick and comprehensive understanding of how repeats are distributed across the entire genome. Accordingly, can it be used to flag potential centromeric regions, which are typically rich and repetitive sequences. And there are 6 steps, as you can see from the workflow diagram. And first, we divide the genome into beans, and for that, we use a back2 and to segment the entire genome into 100,000 KB beans. And second, we annotate the repeat with a window masker. So window masker is a relatively old tool. You might ask why? Because we need it to be rapid. We tried the repeat model and repeat masker, but somehow I was struggle with how we put in this repeat annotation in the beans. But window masker is perfect in this case. So window mask, as you know, is tool designed to identify repeat sequence in a genome to annotate all the repeats present across entire assembly. So then the next step is assign repeat to the beans. So remember the first step, we divide the entire genome into 100 KB beans, and then we window mask to mark up the repeat, then we put them in the beans. And then the fourth step is to clean and the reformatting and prepare for counting. And the fifth step, and once the data is clean, we use the BET tools map the repeat density value back to the original genome. So each repeat would have the original genome's coordinate. And it's the same approach as the read coverage. We would like to visualize the repeat density and using BigWig file. And therefore, the last step is to convert into the BigWig using backgraph to BigWig. 5 telomere, which is straightforward and very simple, and is telomere annotation is essential parts of the genome curation, as you know, and it provides a valuable information about the structural organization of the genome. By identifying telomere, we can pinpoint approximate location of the end of each chromosome or scaffold, which is very important to understand genome integrity and structure. So as you see, the first step in 5 telomere workflow is to determine the specific telomere motif. There are quite a few telomere identifier, and you can gather. And in our so we also have an indoor build tool, which is under development. So I'm currently writing it in the Rust. So once we have the telomere motif, and we use the 5 telomere tools and developed by a person who used to work for Earth Biogenome Project, Sergey Korent. And so this tool to scan the genome and locate the telomeric regions, and it works to search for telomere motif in 1,000 base pair windows across the genomes. Any window that contains the telomere motif is annotated as telomeric regions. And this process provides a clear, precise map of where the telomere are located within the genomes. So that's the 5 telomere. And the last metrics I would like to talk about for the rapid curation is the 5 gap. As you can see, so 5 gap workflow is even more straightforward than the 5 telomere. Is a very effective method to locate a gap in the genome assembly. It uses a tool, again, so developed by Lee, got 60K, and typically it's a content function. So the function scans the genome for region where ends appear, which typically represent the gap in assembly. The workflow then marks all the regions with gap and record their position. So after running the content functions, the workflow converts the gap data into bed file format for visualization. And so that's a 5 rapid curation workflow. And next, I will go through a few we use for the full TreeVal curation. Gene alignment. So gene alignment is one of the complex workflow within the TreeVal bundle. And when, as you know, when we're working with more complex genome, as Will mentions, we often need to gather extra evidence to make accurate corrections. The rapid curation might not be enough, and like when we have to look at the bird's microchromosome and amphibians' repetitiveness in the genomes, and gene alignment would make huge help for these curation purposes. So yeah, we use the 4 curations for various projects in the past, including the Earth Biogenome Project I just mentioned, and the Genome Reference Consortium (GRC), and also the project Will mention, and ASG and ERGA, etc. And we did apply for curation for that. So as you can see, the gene alignment already pop up on the screen, and it divided into 2 major parts. And for the first part on the top, and as you can see, is nucleotide sequence alignment. We align nucleotide sequence like cDNAs and CDS and RNA to the genomes. And we once again use a minimap splice function to do this, and aligning the cDNA, CDS, and RNA sequence to the genomes. And to make the mapping more efficient, we split the cDNA, CDS files into smaller chunks, as Will mentioned earlier. After the alignment is done, then we merge the result into a single output using sum2merge. And then we also convert the merge bound file into the Bed12 format using Bed2 bound to bed. And then the next step is to create a big bed file. So some of you may already have experience with big bed. Is another popular file format, and you can use for the visualization on via view of the genome browser. Part 2 at the bottom highlighted here, and is a protein sequence alignment, and is actually a highly recommended step for genome corrections when annotating genes according to different species. And yes, our curators do highly recommend to use the protein alignment rather than the nucleotide sequence alignment. So because they found that the protein alignments is actually helpful for making accurate corrections. For these, we use a mini plot to build specifically and for protein sequence alignment. So just like the nucleotide sequence alignment, we split the protein FASTA file into smaller chunks to speed up the process. Mini plot, sorry, I forgot to mention the debugger. So Sam again is Henley. And so thanks Henley that he generated this very helpful tools. And yeah, so when we implementing the gene alignment, he also provides a lot of help and lots of help in look at the data and help us to define what kind of parameter we should use for the gene alignment. And so the last extra features of the gene alignment workflow is the ability to generate punch lists as the read coverage. And these punch lists are mainly used for evaluating duplicate genes, and they help curators to quickly spot and fix any genes duplications, which is crucial for ensuring that gene assembly is both accurate and complete. And CellCom. So CellCom stand for cell comparative study, and the workflow and holds a special significance as is primarily used for assess the haplotypic duplication. In the early stage of ensemble analysis, CellCom was also utilized in defining chromosome structure. Inspired by the ensemble approach and drawn our curation experience, we have redesigned the cell comparative workflow to fit the modern needs. And I'd just like to quickly go through the CellCom workflow. And first, we need to do the genome segmentation. So divide the entire genome into equal segments as about 50 kb in the length. And second, we then do the one-to-one alignment with the MUMA. So I'm not going to go through MUMA. So as you know, MUMA is a very decent aligner and provides a very in-depth and detailed alignment and while using it. So we use the MUMA to do the contig-to-contig alignment. And one very important aspect to the note is that the CellCom process is very memory intense, and to optimize the resource usage, and we limit each comparison to 1 gigabyte of sequence at a time, allowing us to better control the memory allocation and avoid excessive resource consumption. And then after we run the MUMA, and then we concatenate all the result into the BET format. And because we have to dissect the genome into small beans, and then we need to convert the coordinate back to the original genomes. And after that, so we do a very important step. So I'll explain why we do that. It's called chaining. So you might be very familiar with the training approach, but in our case, it's different. So once we have the contig laid down, and then we use so I develop a tool and that to chain together all the aligned contigs, allowing for up to 100 kb index and in the alignment. So this training process helps to create a set of reasonable alignment trains that present meaningful relation between the different segmentation of the genomes. And then in the final step, we integrate all the results into a cohesive output. So it provides the curators with a clear picture of the potential haplotypic duplication and structural features within the genome. As you can see, there's 2 bars, which is at bottom of the presentation. And so this is a typical event when you see the haplotypic duplications in a genome. So once you see the 2 alignments, so after training, the 2 chain, they next to each other and neighbor to each other, that might well be the duplication event. Pascal analysis. Most of you run Pascal. In our case, the Pascal analysis is a bit different. So because we have some indoor build algorithm for it, and our initial motivation for developing the Pascal analysis workflow was to examine the distribution of butterfly ancestral unit within the modern butterfly genomes. And this workflow only executes when a ancestral unit table is available. And first step, of course, is to run the Pascal on the genome of interest. And second important things in this workflow is you should have an ancestral unit table. This is a key input for the workflow. And we have one, and many for, as I said, is for the lepidopteran ancestral unit. And it is created based on research into the chromosome structure and evolution in butterfly and moth. So this research was conducted by Charlotte Wright, and butterfly experts from the Tree of Life project, as a part of her PhD work. Her study focused on using ancestral unit, marine unit, to assess the chromosome structure and fusion events in various butterfly and moth species. The ancestral unit table, which she generated, serve as the primary input and for our ancestral unit analysis. So once we have the Pascal gene annotate in a full table and also the ancestral unit table, then we map this ancestral unit onto the genomes of the lepidopteran species that are of interest. And this mapping allows us to visualize, assess the distribution of ancestral chromosome structure within the genome. One important note here, so we are planning to extend our Pascal analysis workflow with other group of ancestral units. Currently, we have a Nigon for nematode and also a bunch of beetles ancestral units in hand. We would put that in our future development plan. And in silico digest workflow was originally designed to compare results with the Bionano de novo assemblies, which is highly useful for genome curation when Bionano data is available. By identifying the enzyme cutting sites, we can determine if there are potential issues with the genome assembly. The workflow is relatively simple. It uses the genome smart FASTA file to directly map the enzyme cutting sites associated with the Bionano data. The location where the enzyme cuts provide insight into structural integrity of the genome assembly and helping to flag areas that may need further attention. So, but Bionano data now we're not really using in the Tree of Life project. So even though with that missing and with the absence of Bionano data, we still run in silico digest workflow regularly. Typically, we can use the 3 different enzymes at once, allowing us to observe the distribution of cutting site across the genomes. And these are 3 cutting enzymes we use is DLE1 and BSS1 and BSPK1. And I hope I think most of you are very familiar with Bionano cutting enzymes. And Synteny, very straightforward, just a straight line from the FASTA file and to align minimap. So in Synteny workflow, and some of them might be very familiar with, you might do training, but we don't do it here. And straightforward, we just use a minimap to simply align genome to genome. So in our DPO ASG VGP projects, we don't assess Synteny for all the species, as Will mentioned earlier. Currently, we only evaluate bird genome, as we have a 3 very high accurate birds reference genomes. They are Gallus, the chicken, and Aquila chrysalis, that's the golden eagle from our 25 genome project, and Tanio pigea gatata, that's the zebra finch. And these genomes are of very high quality. For Minimap, we use the parameter -XASM ASM 10. So which means that we assume around 10% of sequence divergence between the query and reference genome. This is particularly helpful for highly divergent and error-prone assemblies. I think that's all I want to cover about the main workflows of TreeVal. And next, let's take a look at how results from this workflow are visualized on different browsers. And first, let's look at what we use for rapid curation and HiGlass. So HiGlass was the first for user genome assembly curation about 5 years ago. So when I only started work on the genome assembly analysis. And before that, visualization of the curation method, metrics rely on entirely on the TreeVal. And my colleague and I, we integrate the entire Hi-C mapping workflow and were the first one to display the Hi-C heat map on the HiGlass. So over time, we gained practical experience, which led to develop the rapid curation protocol. As I mentioned earlier, over about 90% of our curation work now for DTOL project is conducted using rapid curation methods. As you can see, rapid curation only requires a Hi-C heat map, read coverage, read density, and telomere and get track. So TreeVal directly places this rapid curation result in Hi-C files folder, as is shown in the red box here. We now have a dedicated Hi-C HiGlass server for DTOL. When the pipeline generates output, the result are automatically loaded into the HiGlass. The entire process is orchestrated by the pipeline orchestrator, Perfect. Okay, we have a fire alarm here. I think it's Wednesday.
Do you guys want to mute yourself for a minute? Right, apologies. We forgot that Wednesday is fire alarm test day at Sanger, which is awkward.
Wednesday.
It's back now. We're back now.
Yeah, so Wednesday, 11 o'clock. Sorry about that. And so visualization part 2, we use a JBrowse. Both me and Will has mentioned that. We use JBrowse alongside with HiGlass to present the full results of a TreeVal. On JBrowse, we primarily display the result of a cell count, in silico digest, gene alignment, and synteny workflow result. And while we also include the result that already show on the HiGlass and Hi-C map, but in the JBrowse, so we use the juicer Hi-C, which JBrowse doesn't allow other Hi-C map to be visualized yet. So we use a Hi-C. That's the reason why we still generate juicer Hi-C during the TreeVal. And that's one of the reasons. Second reason is that people would like to use a Hi-C externally and from the community. And so when you run TreeVal, and then you'll find the files needed for JBrowse are located in a folder called TreeVal upload, as indicated by the files marked in the blue box here. Yeah, so punch list. We mentioned punch list twice while I was going through the workflow. How we use a punch list. And the punch list could help you quickly locate a problematic area in the genome. In JBrowse, we output the punch list as a BET file, and JBrowser's spreadsheet feature can read this BET file. And when you click the chromosome coordinate, which is here, you can see they are in blue. When you click them, it will navigate you to the corresponding genomic location. Right, so this slide in the green box, it holds some text file created by TreeVal Nextflow pipeline, like the execution log, pipeline diagrams, and execution files to help us to get better understanding of how TreeVal runs. And Damon, the chair here, and he put together a groovy model that gather information about the input data. the goal here is to see how the input affect resource allocation in one time, so we can optimize things as the TreeVal and the DTOL project goes.
Can I just jump in and explain just a little bit more on that, please?
Go on.
So at the minute, I have a couple of mentees which are utilizing this data to produce in a machine learning model, which will then be able to dynamically assign resources as the pipeline goes. So at the minute, this is very TreeVal specific, where in the future it is our want to make this a very generic model so that all pipelines would be able to dynamically assign resources as they go. Yeah, that's it.
Thanks, Damon. Yep. I think I have to wrap up from here. I will hand back to Will, and he will guide you through and how we get on with the practice in report. Thank you.
Can we just answer a couple of questions first then, please?
Yeah, sure.
Right, so there's a couple that I haven't answered as of yet. So another one from Sam. The basic principle of gene alignment is not to make a heat map adjustment on high coverage protein, cDNA, RNA alignment, correct? Yeah, I don't think the curators are making changes on the heat map based on information from the alignment, but not
Yes.
exclusively.
Yeah, so currently our curators are mainly looking into a punch list. So gene alignment and give us quite big numbers of punch list. Usually, and what they do, if you have opportunity to join the menu curation sessions, and yes, so they one of the ladies, she's an expert, Jo Collin, and she will give you more insight into that. So as I know, and they first picked up the punch list from read coverage, from where and the duplicate the potential duplicated event where that will combine with the gene alignment punch list. So it's they're not only looking at one set of metrics, but combine the others events as well.
Cool. Thank you. Then there's one from Terry. Long read assemblers output 2 pseudo haplotypes. Can you comment on how this is handled with TreeVal, please? Do you run both separately, or is there an option to run both together? I've seen both pseudo haplotypes curated together in pretext.
Very good questions. So recently, we changed our strategy, and well, the ideal world is we work on single haplotype genome. So, but the real world isn't that simple. And so what you see the two haplotype together, two haplotype together, and it's because they want to see the behavior of our upstream purging if that work. So we combine these two haplotypes together, right? So then the Hi-C map will look quite different. You will see some in parallel signal alongside with the diagonal line. And this is very important for the curators to actually flag out if the duplication event, if the haplotypes still remain in one or the other.
This doesn't take place within TreeVal though. We have to prepare the combined map beforehand.
Yeah.
Combined assembly beforehand.
So we don't do that in TreeVal. We do or we don't do, but we do combine two FASTA file of the two haplotype and run through the entire TreeVal process. So that's how we handle the do-hap in TreeVal currently.
I imagine they will show an example in the manual curation session, to be honest, because quite a few of them come through as combined tickets now.
I can show a quick example as well this session if anyone interested.
Yeah, go for it.
Right.
Yeah, should I stop sharing and can show you the nice map?
There's another question from Sam as well. Any thoughts on pair tools for generating dot cool, for example?
I use a pair tool, and it's basically pair tool is a part of my pair tool. Okay, so I create the contact pair based on the pair tool. It's one of the components, but I did a lot of filtering after the pair tool. So it's no longer pair tool in the TreeVal, but it's called the Tree of Life pair tools.
I haven't got one to hand at the moment, but I can prep one for after the break.
Yeah, sure.
Can I just? You're not sharing?
I'm not sharing anymore.
Okay, before we go through the demonstration, I'll just quickly run through a list of pros and cons for the TreeVal, most pros. Generally, the main criticisms of TreeVal or difficulties we've had have been relating to anything that would affect any other workflow language, really. There's been some issues with error reporting and locating errors, but both of those have improved significantly in Nextflow whilst we've been using it. So I'm hopeful that that'll continue to be the case. As a result, their standards still being development. We've had to update process to keep in line with that a few times. And also Nextflow has ended up having a little bit of a steeper learning curve than some of the other workflow languages. But we've found that the advantages it gives have been worth it. Now, the pros for TreeVal. It is a singular pipeline for all the data curation generation. It has a higher and quicker throughput compared to the old, say for example, compared to the old GVal pipeline. One example was a lepidoptera genome would have taken about half a day. This around 500 megabytes. With TreeVal, we see it at least twice as fast. We can even get some small genomes like nematodes down in under 30 minutes. We're no longer relying on a database using a set of flat files in a set directory structure, which means it takes up significantly less storage space. For example, the cricket, which is 1.6 gigabases genome, sorry, let me just pull up the stats. All right, yes, under GVal, the storage requirements for the cricket were over 1 terabyte. Though under TreeVal, it became less than 50 gigabytes. So with this massive storage shrink, the only thing you have to bear in mind is that the intermediate files can end up being quite large, so you have to have disk space available, but they can be cleared down immediately afterwards. Hopefully, that will no longer be the case soon, as Nextflow are planning on having intermediate cleanup steps. The purpose of writing pipelines in Nextflow is for the democratization of science. Nextflow pipeline written properly with relevant conda containers and config files being referenced to be run anywhere with sufficient resources. A single mid-range laptop, including upselling size of local HPC, can be used for running the same pipeline. This hardware agnosticism is a major benefit to the TreeVal pipeline and Nextflow pipelines in general. A good demonstration of this is the fact that we're able to run it on Gitpod, as we'll see shortly. We are able to configure it to a very high degree because the resources can be managed at a process level and also can be managed dynamically so that retries can calculate updated resources. We've also been able to prototype it very effectively by pulling out individual processes and running them individually, which has been a very useful aspect of using it writing in Nextflow. Yeah, we found Nextflow and particularly NF-Core, a very effective, very useful tool set, and we believe that everyone should use it and therefore expand our existing TreeVal project. We are planning to maintain the TreeVal project for the duration of at least the detailed project, so we will be constantly looking at alternative softwares and other data to improve curation and adding that into the TreeVal project. Okay, now we'll come to actually running it. Has everyone everyone needs to go to this link here, the BGA Academy session documents TreeVal tutorial, which will give you the link to send you to Gitpod. It should look something like this.
This is also the same page that's on the GitHub repo as well, just so people are aware.
Okay, so yeah, to start, to launch it, you want to press the send me to Gitpod. Once you reach, I presume you have to essentially sign in with your GitHub account at this point, usually if you haven't accessed Gitpod before. But when you do, you want to be able to open a workspace using the default VS Code browser and standard resources. Can people raise hands in the chat when they have been able to access this? It may take a bit of time to load the container image. Great, quite a few you've gone through to Gitpod. If anyone's having any specific issues, please put them in the Discord channel. But you should be able to see a page like this. Gitpod allows us to run all of the different softwares just as in a sort of demonstration purposes. So all of the relevant tools have been installed on startup. There's still quite a few people who haven't raised hands. If you're still having issues accessing. We're taking a break shortly after this, where we can resolve any issues. This is just to get ourselves started. Okay, great. There's quite a few people who are through now. So yes, if you have gotten through to this workflow page, the first thing we want to do is to be able to run the next flow, which is all very simple. On the tutorial page, there is an example command here. This is for running the example BGA test YAML in rapid mode. So this should complete. This potentially will complete within, how do you reckon, Damon?
Hopefully quick-ish.
It should hopefully be quite quick. We don't need to rely on it completing though, because there is data stored on the Gitbod image anyway for you to have a look at. But it's quite simple running Nextflow. Just copy this example and run it in the main window. So that's a clean bash window. Sorry, not that. That's just various components finishing installing. Having an issue with. There we go. Okay, so you should see something like this. Okay, so we should take a brief break now while that runs, and in that time people can message to say if they're having issues, and we can try and help you get to this stage. Is that all right with everybody? So we'll aim to resume the tutorial on the next stage in about 15 minutes. So 20 past 11. So feel free if it's running okay for you, feel free to step away, basically, for 10-15 minutes.
And don't worry about all the warnings that pop up initially. That's just because we're running the rapid workflow on the pipeline. The full workflow has a lot more processes going on.
Yeah, these
just
are
letting you know that all the full processes are not being found in the rapid run, so it's throwing a warning. It's nothing to worry.
self-comp, gene alignment, synteny. So yeah, the full processes. So you see each individual process is being run, and as they complete, you get a little tick. So is anyone? So have we got anyone with any issues?
Oh, looks like we've got a couple of pods where things haven't installed properly.
In some cases, it might just be a case of letting installation fully complete. If the next flow installation is this bash window, so check that this one is fully completed. Yeah, so yeah, trying a restart might be the best for some people.
Choosing the white workspace. Just the initialization is it failed.
So Terry's resolved it from refreshing. Connie's resolved it from refreshing.
Yeah, I think we're in an okay spot now.
Yeah, Afia Cheetah who's having the same issue as Terry B. Have you been able to restart and have it working now? Great, thank you. Yes, no.
It's quite nice watching it run on Gitpod. We didn't get this far last year.
Yeah, it probably won't finish by the time that we need to be looking at data, but it's nice seeing it in progress. Yeah. This is a stripped down nematode that we're running on, aren't we?
Yes, this is a very heavily subset nematode data set.
So should finish within half an hour or so or less.
Yeah, I mean the fact that yours is already done the mini map. Oh no, it's doing the the supermodule. But yeah, shouldn't be too bad.
Yeah, so although you can run TreeVal quite happily on an individual laptop, for getting the resources required for larger genomes, we definitely suggest putting it on the cluster, putting on a HPC cluster.
Right, we don't actually do any annotation whatsoever, if that's what you mean, Sam. Yeah, this is just purely we're just aligning the gene set data to the genome and leaving it at that. We're just using it as evidence. I think there is an idea going on at the minute that we should start doing our own automated annotations, but that's not something that is going to happen particularly soon.
Yeah, the data set should be available, should be viewable in the file browser up here. You should be able to see whether or not that file is available.
Oh, interesting. If you click through the tabs on the right side of the terminal, we should be able to see whether something has failed or not.
Okay, it's 20 past, so I'm assuming people are back or getting back now. So we shall move on to the next stage, which is viewing data in JBrowse. Before we start, there's just one bit of manual setting up that's required. On the install JBrowse 2 serve tab, you basically just have to type Y and allow it to continue. And that will complete the installation and make JBrowse available to view. Okay, JBrowse, as we've already discussed, one sec. JBrowse, as we've already discussed, is a actively developed genome browser built with React and JavaScript, which has been proven to be easily extendable due to the extensive documentation. There's also a number of other related tools, like a desktop version, but we're using the browser version at the moment. This example here, you can see is a JBrowse viewer lepidopteran showing a subset of data related to ancestral and standard buscode coverage, as well as peptide and gene set, cDNA gene set alignments against another lep. So the top there, that's the coverage track, and then the ancestral regions track, the buscode gene, cDNA, and peptides. Underneath is a list of the file types natively ingested by JBrowse 2. Though, like you said, this is potentially extendable by writing your own plugins. Right, now to add one of the files that we've produced into JBrowse, we use this command. We've provided a list of the commands here in the tutorial that you just need to copy and run in a new tab. When it opens. So as you can see, these are all being added, add tracks added with these names. It's the config.json that is behind the JBrowse. Please post any issues in the Discord. Then to view, you open up JBrowse Web by going to ports and pressing the external open browser button here. Sorry, I might go through that a bit more logically just to make it clear. Open browser, linear genome view, select the lepidoptera, open, and then you press this button here to make visible any of the tracks that you wish to look at in more detail. Is anyone having any issues with opening JBrowse?
So could they load the result while the process is still going? Should have the result in,
No, this isn't using the result of the process. That was just to demonstrate.
yeah.
This is a Lepidoptera that was already loaded onto the Git pod.
So the question is, do they have to wait for the next flow process to finish and then go?
No, the next flow process you're running was just to demonstrate how to run next flow. That's using a subsetted nematode. This is a Lepidoptera data that's stored on the Git pod for illustrative purposes. So you don't need to wait for it to complete to be able to open up the JBrowse.
Can you quickly show how you set JBrowse up? I think there's a couple of people that haven't got it running.
Okay. Well, from what step?
So in the install JBrowse 2 tab,
Oh, right. Yes.
people go there. You may have to tell it, yes, I want JBrowse to be running.
It might say, okay, to proceed, yes. In which case, just type yes, and that creates it, sets it running. To add the tracks, you open a new bash window and copy and run this block here, because it has all of the add assembly add tracks. And then once it's available, you open the browser by going to ports and then pressing the open browser. And then you can access it by selecting linear genome view, and that's all available.
Well, could you do that again? Kirsty just asked questions. Which link to press to open JBrowse 2? Just exactly what you did before.
This button here.
Yeah.
So on ports, the top port is JBrowse Web, and you want to click Open Browser, which is a little globe.
Okay. I'm still confused. Where are the JBrowse command being input?
JBrowse commands and just go to any, just open up a new bash window. So just open up by pressing the plus button and then just run this in when this finishes loading.
You have to copy the command
from the tutorial.
Yeah, from the tutorial.
So from this page.
If you cannot find the page and if you in the Zoom chat, it's on top of the Zoom chat. The link.
It should also be coming up in the Git pod for, yeah, that simple browser one. That should be pointing to that same web page. How's everyone doing now? Are we all able to load it up? Cool, fantastic.
Okay, has everyone's managed to access
Can you go through
JBrowser?
the process of opening JBrowse again, just from opening the browser and selecting the tracks? Oh, okay, no, never mind, they've got it. Carry
Okay,
on.
great. Okay, if everyone's happy with opening JBrowse, the other one we want to look at is opening data in HighGlass. Once again, there is a section on the upload to HighGlass. So you wish to copy these commands, which will ingest the data into the HighGlass browser. And again, on a fresh bash window, run those in. Now this might take a little bit longer. Let's get in there. So it's ingest the CUDA file, ingesting the genome file. Okay. Mine is completed, yours is probably still going, but for this one it's much the same. Go to HighGlass, open browser, so open HighGlass. Okay. It's finished loading. Sorry, the other one also. When it's finished loading, press the plus, center, and then it'll take a moment, but it should identify and find the available files maps here. Here we go. So you want the map and the genome track.
Okay.
What?
Shouldn't click the genome track at the same time because the grid, the genome look is behind the thing now.
Yeah.
So if you close it again, close it.
That's true.
Just close the two sessions.
I'm leaving those sessions open because.
Okay.
So.
MCool first. Then the genomes. Now you can see the scaffold boundary. The grid is on top of the map.
And to load the additional curation tracks, you go top, TreeVal test, and then you can include any of those you wish to.
You can expand the top tracks as well. So if you, and if you right click on one of the big tracks, the settings, and you can change the style of it. Track type, then change the track type to lines and points, anything that you prefer. My high glass is empty. So how empty is that? Have you Sam? Have you add a track on, like what Will just did?
Please do the high glass opening steps again.
And you have to open the high glass browser.
So after running it into a fresh bash window, you want to just do the same process. Go to ports, high glass, opening browser. You should see this window. Press the plus, center. Then you should be seeing, let us know if you can't see a track here. It might be that it takes a bit of time to load in, but then you should see this TreeVal test option, and from there you can select MCool and submit that to open the heat map.
If you had JBrowse open before loading everything into, if you had high glass open before loading things into high glass, you'll need to close it down and open a fresh high glass, because it'll like caching and whatnot.
Sam, have you got the map on the high glass now? Okay.
Oh, good.
Oh, thanks.
Cobus, are you okay? Do you still want me to go through the loading the tracks again? Oh, great. Yeah, so once you've got the heat map open, it's a case of going and adding track. Doesn't have to be top, it can be any position. But if you go top, and then you should have the other tracks available to load in.
Yeah, I'd like to mention one thing here. For the bigwig chart, you will see on the left, and you will see a label, right? So for both the repeat density bigwig and the coverage bigwig. But unfortunately, the high glass is not able to label the bed track. So you can see on the top, that might be the telomere track, and the third line, and it can be the gap track accordingly. So it doesn't have the label, and you should remember and what you load first.
How to fix that?
Yeah, how do you fix that?
It's just in how when you're loading the tracks, it's a different value you have to set rather than
the name.
No, it's not the name. Yeah, it's going to set a different label. I can show you later.
Okay, right.
So is everyone okay now? Okay, great.
Cool.
So yes, the curator. So yes, we definitely would recommend, if it's still open, I believe it is, attending the manual curation talk. So, excuse me, as they'll go into a lot of detail around using high glass and TreeVal's full manual curation.
Do we have a pretext installed in the report?
Do we, Damon, have pretext? We do, don't we?
Yes, we do indeed. So if you go to ports, oh, actually, no, if you go to terminal first, in your, so there's an install curation pretext there. So if you go into a new tab and do // view space ampersand.
Can you post that command in the chat?
// just type
view ampersand. It should then come up with a little "ooh, I'm running" sort of note. Yep, there we go. Fantastic start. Did it in, oh dear. Okay.
While Damon's looking at that, I'll just quickly, for someone who was requesting it earlier, I've got an example of a single app and then a combined map for that same app type earlier. So select that's being looked at in the curation. How long do you need for Zippy and the curation preset?
5 minutes.
5 minutes.
Even 3 minutes, I think.
See if we need to.
Do you have a pretext view in your installing local?
Local, I can show it.
Yeah, and just in case couldn't find it, pretext view is very important in terms of the manual curation, rapid curation.
Yeah, they'll go into that in more detail in manual curation, of course, because for the actual manipulation of the high glass map will be done through pretext rather than high glass.
There are instructions in the wiki. Which I will have to replace. Yes, and so there's some recipe and from the genome informatics team, they have a page. If you join the session, and they will introduce that to you.
Right. I think the issue is I didn't install pretext.
Okay, that's fine.
So, I mean, I'm going to post the commands in to do it. It won't take long at all because it's just that one file. So if you want to run pretext, you can execute that command and then the ./pretext view ampersand should then work. There we go. So then if you go to ports 6080 and don't make me a liar. There we go.
There was a question in the chat, is if the TreeVal is already running rapid curation mode and I want to run an additional module from the TreeVal, from the full mode, but not all the modules in the pipeline. For example, only the classical analysis, can I do that? Answer is yes, but not on the current release. So we had done some further development, and based on the release that we have on the GitHub at the moment. So all the workflow can be run individually or collectively.
Yeah, so I think at the minute it's set up the new version, which will be releasing at some point. There will be an exclude flag, so it's more you exclude sub workflows you don't want to run at the minute. However, if anyone has thoughts on how they would want that better implemented, please open an issue.
Okay, this is what the pretext view look like. So
Do you know which the pretext file is, Damon?
It's in if you mount it already, it should be on the Tree of Life server. You're not on. Are you local or you?
No, it's
okay.
on the Git folder.
Okay, in that case, ask Damon, and should be in the same work IC folder, right?
I found it now.
Okay. So that's the same butterfly, same moth. Sorry. This is a map. How does it look on the pretext compared to high class? And as you can see, is the resolution. It's okay. So very clear diagonal signal.
But yeah, well, we haven't got time to go into it in detail here, but this is what the curators use for actually manipulating the pretext map for curation purposes. And that's all achieved through this desktop tool.
Can you try some of the...
Yeah, can you do some cut and join, just like rock to move? roughly
I'm trying
move
to
there?
remember the shortcuts, to be honest. What's edit
It's
mode?
E, just E, I think.
Yes. Thank you.
Just drag it up to the top. Yeah, that will do. So
It runs a lot better not on Git pod as well.
If you
There we go.
click
You can see
on the
you can
mouse
see a
as
region
well.
being moved there. So that's
If you
click to
middle
region.
click, it'll pick up the whole chromosome or the whole scaffold even.
yeah, it's okay. It's a terrible move, but so that this will tell you you can move the content. Yeah, we don't have the curator's golden finger, you know.
Yeah, no, we don't. I'm not practiced with using brew text at all. So no
Yeah.
shortcuts.
Yes, then after that, and if you're very satisfied with what you have done to the predicts map, and you could output a AGP, assembly genome structure file from somewhere.
Oh, I made a right mess there.
Control U, then you can bring up the menu. Yeah, generate AGP. Then you can have the AGP file of the this state. And so that's basically the record you should keep, and also some roadmap and records and tell you what you have done to the pretext map.
Can you try opening some of the tracks that should have been loaded into this pretext, please?
Doesn't look like they were.
Extension. It doesn't have the extension.
No, okay. Normally the tracks for gap, telomere, coverage and whatnot are ingested into the pretext, so you can see them on both the high glass and pretext map, which is very handy for the curators as well.
It's like playing jigsaw. It's very
Yeah.
It's
very skill. Okay, so we've only got just over 5 minutes left. So do a quick, Yumi's going to quickly go through Zippy pretext and curation pretext.
Okay.
Just stop sharing.
Yeah, I hope you all had a very good practice and have an overview and for the how to run the TreeVal and how to display the result. And alongside with TreeVal, we have some extension or accessorize and for people to use and after curations. So pretext, the curation pretext is a smaller version of a TreeVal, I'd say. So once you've done one round or second round or third rounds of curations, and you want to have a very quick overview about the work you have done, and you can run this process and to generate pretext map, not to the high glass and high C, juicer high C, and etc. So it doesn't really include the full TreeVal analysis, but only the rapid and up to pretext mode. So this pipeline has two entry points, or map, and choosing or, and we'll run the sub-workflow to generate like gap, telomere, coverage, and repeat density data, which can then fed into the pretext we just showed and display the result. The map-only version, and on the other hand, will only produce the pretext map and snapshot. So that will give you a quick overview and what your curation work look like. As you can see, the workflow is rather simple compared to what I showed you before with the high-C mapping workflow. So it's only up to the pretext file get generated and everything stopped. And I would like to mention another development within Tree of Life. It's called Zippy Pretext, as its name suggested, and it's zippy. It's a way to handle large genome curations. So why we needed that? And remember, at the very first beginning of my presentation, the first slide about high-C mapping, we suggest you to save the alignment file in the binary format, and from the YAS actually. And so I would like to show you the Zippy Pretext workflow. It's a handy tool and developed by my colleague and I, Chen-Si and I. And so occasionally in your curation work, you might meet a larger genome. You don't want to run the remap because remapping. So remapping by Minimap or BWA is very time consuming. Let's imagine that you have a 100 gig mesotome. This is a larger genome, so largest genome we curate in Tree of Life, or a 25 gig newt. So the mapping would take a substantial amount of time. So if you, in the first round, you saved the bin file, the boundary file from YAS, and keep your FASTA file on the side, and then you can run this process. So this process is straightforward. First, you run the script, rapid join. So sorry about the name. The rapid join doesn't really join anything, but it produces a TPF file, is another chromosome structure file, and from the previous reference. Then you use a pretext to TPF, a tool created by my colleague James Gilbert. And so these tools convert AGP file from the pretext map you've just seen to a proper AGP file. So why we have to do that? And pretext view is written in C++, and especially using the OpenGL library. So all the image, they are textual based. So the coordinate and from the pretext AGP doesn't really reflect the true coordinates of a genomic location. So this gentleman, James Gilbert, and he kindly offered to write a tool to convert all the pretext based file into a proper chromosome and related the actual genome coordinate related files. So once you have the corrected AGP, so output from the pretext to TPF, and then you can use a user pre to create a curated alignment.txt. So if people have practiced with user pre, you probably know this step well. So the alignment.txt essentially is just like the pair file, and which I think Sam mentioned, or Terry, I don't remember in the chat, is it essentially look like a pair file, and from the pair 2 as well. Then you use a pretext map to take in this alignment txt, and then produce the new pretext file. So by our practice, it could significantly saving the mapping time and for the large genome. And so if you are a large genome collector and you like to curate large genomes, or you already have a large genome in hand, so do have this in mind, and or contact us and how to use this tool, ZB Pretext. So I have a bash version of a pipeline which is already available, but the ZB Pretext is under development into Nextflow. And I think that's it. What I want to say about the other utilities we have in the Tree of Life project. I would like to quickly wrap up with the acknowledgement. So thanks to the Genome Reference Informatics team, short for GRID. If you have been visit to sign up GRID team, they provide a menu curation guideline. And our team is a Tree of Life assembly, and also the Tree of Life infrastructure team. They help us to build up the Nextflow infrastructure and high glass server, JBoss server, etc. And also thanks to NF core and community to guide us through and from the very beginning and how to write the module and to the end how to put all the workflow together. JBrowse team and high glass team, they are very, very helpful since day zero, and to teach us hand on hand how to display the different data type. And special thanks to several persons, including Goyin Chi, Matthew Rapoto, and from the Tree of Life infrastructure team, and Joe Wood, Sarah Pellon, and Joe Collin, Michael Polini. They've been helping us to validate all the track information come out from the workflows. And Charlotte White, and we mentioned her before, and she's the one who delivered the Lepidoptera ancestral unit table. I think that's all, and thanks for joining today. I hope you're enjoying learning how to run the Nextflow pipeline, not only for TreeVal, but in the future you might meet some other workflow which is useful for your research work. And you are very encouraged to contact us and through email, and which is I put on the slide here. And also if you can, if you have other idea and you want to incorporate into the TreeVal, and then you can open the issue on our GitHub. And you are encouraged to join the Assembly Curation Slack channel if you have any curation related question to ask. So thank you for joining today.
All right. Thank you both for running the session. Does anyone have any questions, please? Feel free to unmute yourself and shout at everyone. That's fine.
So the TreeVal, actually, I would like to add something on as well. So we designed TreeVal and workflow is mainly to reproduce the analysis we did for GRC, for VGP at the very first beginning. And then we select a handful set or multiple sets of a workflow, which we believe is very helpful for the menu curation. And there are other recipe, for example, linkage map, genetic map analysis. So we have a run through that as well. And we encourage you, if you have any idea and how to improve the menu curation, so we could exchange idea on the Assembly Curation Slack as well. Any question?
I haven't seen any questions.
Okay.
Just a lot of happy viewers. Fantastic.
Great, thank you. I think we up to the time now.
Have a good rest of BGA for those of you go attending other